Welcome to this explainer. Today, we're diving into what is arguably one of the most mind-bending questions in modern biology. I mean, we're looking at a frontier here that honestly feels straight out of a sci-fi movie, but it's rapidly becoming science fact. The big question: can scientists actually create life? We're going to unpack the cutting-edge research, look exactly at what we can and, crucially, what we cannot do in a lab today, and explore the wild future of biological engineering.
So, can scientists create life? This question really sets the stage for our entire journey today. We're going to explore this massive gap between what we can build right now and that ultimate biological finish line. It's the kind of question that completely challenges our everyday definition of what it even means to be alive, right? Now, scientists have made some truly incredible progress creating lifelike systems, artificial genetic materials, and engineered organisms. But, as you'll see, conjuring up a completely new, independent, living organism entirely from non-living chemicals, yeah, that remains one of the absolute greatest unsolved challenges in science.
But before we look at what scientists can do, we kind of have to establish the baseline rules of the game. Like, what actually makes something a living organism? Because life isn't just one single thing. It's more like a dynamic checklist. To be considered truly alive, a system has to maintain a metabolism. Basically, it needs to use energy. It has to grow and reproduce on its own. It needs to store and use genetic information, respond to its environment, and this is the big one: it has to evolve over generations. If you miss any of these criteria, well, you haven't truly created life.
And when you look at those requirements, keep in mind that creating life is absolutely not just about stitching some DNA together. It's about jump-starting this incredibly intricate chemical engine. A living organism requires a flawless microscopic choreography. You need DNA and RNA to act as the instruction manual, proteins to actually do the heavy lifting, metabolic pathways to burn the fuel, and cellular structures to house the whole shebang. To put that into perspective, a single microscopic yeast cell contains over 40 million protein molecules, and they're all just bumping into each other, doing their jobs flawlessly, without a central brain telling them what to do. It is a wildly complex, interdependent network.
Honestly, a great way to think about this challenge is like the difference between editing a book that's already been written versus staring at a blank page and writing a completely original novel from scratch. Right now, science is incredibly good at the editing part. We can tweak and modify the existing code of life all day long, but writing life entirely from scratch, using only raw, non-living chemicals, that blank page is exactly where current science hits a brick wall.
Now, that being said, we are already heavily editing life in ways that directly impact your health and the world around us. Scientists can design and produce artificial DNA sequences in a lab to develop new medicines or study how genes work. We can also modify existing organisms, which you probably know as genetic engineering. Think of genetically modified crops that are designed to resist disease, or engineered bacteria that are specifically programmed to pump out insulin for diabetic patients. We're essentially hacking the genetic information of existing life to solve massive global problems.
Let's look at the concept of a synthetic cell. This was a huge milestone, but it comes with a pretty major caveat. Researchers actually managed to create a bacterium with a completely laboratory-designed genome. They literally wrote the genetic code on a computer, synthesized it, and it worked. The cell could grow, it could reproduce, and it carried that synthetic information. But here's the catch. They still had to insert that synthetic DNA into a pre-existing, natural bacterial cell. So they built the software, but they still had to borrow a naturally manufactured piece of hardware to get it to boot up.
Researchers are also out there building these simplified, cell-like structures called artificial cells and protocells. Artificial cells can actually hold genetic molecules and produce proteins, sort of mimicking basic biological functions. Protocells are even more basic, just lipid membranes and simple chemical reactions. So why does this matter? Well, scientists were basically using these protocells as a biological time machine. They used them as models to try and understand how the very first life on Earth might have formed out of the primordial soup billions of years ago.
So looking at this quick rundown, it's clear we have an unbelievable toolkit to engineer and redesign parts of biology. From synthesizing artificial genetic sequences to designing entire genomes and building cell-like systems. But if you look closely at these milestones, none of them equal creating a totally independent, brand new organism exclusively from raw, non-living chemicals. We are, without a doubt, master mechanics of biology, but we aren't the original inventors just yet.
To understand where the next massive leap is going to come from, we have to talk about synthetic biology. This is the exact field trying to push past these boundaries. By combining the rigid, design-focused principles of engineering with the messy, complex world of biology, scientists in this space aim to construct entirely new biological systems. We're talking custom-built engineered microorganisms, advanced biofuels, and sustainable materials that could literally revolutionize our economy.
And within synthetic biology, the achievements so far are just staggering. Beyond those synthetic genomes we talked about, researchers are also creating what are called minimal cells. Basically, they take a cell and strip it down to its barest minimum, removing everything except the absolute essential genes needed for survival. By doing this, scientists are trying to figure out the absolute bedrock requirements needed to sustain life. They're looking for the absolute floor of biological existence.
And this technology isn't just some abstract science experiment. It has direct, immediate impacts on our future healthcare. For example, scientists can now grow organoids. These are small, simplified versions of human organs grown right in a petri dish. We're talking brain organoids, liver organoids, intestinal organoids. Having these mini organs allows researchers to model complex diseases and test life-saving drugs safely on human tissue long before they ever have to be tested on actual human patients. It's an absolute game changer.
But, okay, despite the sci-fi shock value of growing mini brain organoids in a lab, we really need to definitively shatter a popular myth right here and now. Let me be incredibly clear about this. We absolutely cannot build a human from scratch. While biotechnology can do amazing things like grow human cells, use stem cells, and develop artificial tissues, creating a complete human being from chemicals or artificial components is a hard no. Actually, it is entirely beyond the realm of current science. Not going to happen anytime soon.
This brings us to the biological roadblock. Let's look at exactly why bridging that gap from non-living chemicals to a living entity is so incredibly difficult. I mean, you have to wonder, why is it that we can land a rover on Mars, but we can't mix a batch of chemicals in a beaker and have it crawl out on its own? Well, the answer lies in the sheer, staggering complexity of biology. A single cell isn't just a bag of DNA. It requires thousands of interacting molecules, complex protein folding, dynamic membranes, and highly sophisticated energy systems, all operating perfectly in a microscopic space. Plus, the ultimate bottleneck is self-reproduction and evolution. A true living system has to maintain itself, adapt, and reproduce independently. It's kind of like trying to build a self-driving car that also has to mine its own metal, refine its own fuel, and build baby self-driving cars in the trunk. Creating a system from scratch that can naturally evolve is a monumental hurdle, especially since we still don't even fully understand how the first life naturally pulled this off on primitive Earth.
But looking ahead, if we can overcome these biological hurdles, the potential rewards are simply world-changing. Future synthetic biology platforms could allow us to design biological organisms with highly specific functions. Imagine designed microorganisms that can literally eat and clean the plastic pollution from our oceans. Or picture a world where lab-grown artificial organs completely eliminate transplant waiting lists forever. The creation of advanced artificial living systems really represents one of the most hopeful, powerful tools for the future of humanity.
As we wrap up this explainer, standing right on the precipice of tomorrow's biotechnology, we have to recognize that with this immense power comes some pretty profound responsibility. So, I want to leave you with these questions to ponder. Just because we might eventually be able to create completely new life, should we? How do we guarantee biosafety and prevent environmental disaster? And ultimately, as we unlock the very source code of existence, who gets to control it?